Um, okay, I'm going to talk about the purification of protein using the BioRad Profinia protein purification system. Dr. Kousoulas has a big idea of using different tags and how to purify them. But I'm going to show you the some demonstration of this machine, how to use this one, and also we have two protein used to purify uh, the protein using this Profinia purification system and to immunize rabbit and got the antibody. So this is the Profinia purification system instrument that we used. And this is, it has different components, like here this is the buffer component, and here is touch screen interface. You can monitor and see the purification steps during purification process, and the sample component. Together we can use two samples, sample 1 and sample 2, and here is the cartridge. This is a chromatographic column, and this is for desalting. When we use this sample for purification, during the purification process, after binding, and it automatically directs the protein to the desalting column and then purified. So finally we got the purified protein after desalting. So the another important thing in this machine is here is a flash drive slot. After you have the data saved in the machine, you can transfer it with a flash drive and analyze it, whatever the purpose. Okay, and this is a buffer diluent. So this system has the buffer, and the machine automatically dilute the buffer with the reservoir here. And these are the fraction collection tubes. You have different fraction like flow through wash, wash 1, wash 2, and then you have the protein in diluted tube. So this is a good machine. If you can have the protein in a soluble form, then it is easy to purify the protein. The important thing is to get the protein in the soluble form. And another key features of the machine is this is already pre-programmed, program already saved there. You just need to use, it has two different systems for the tags, one for GST and another for his. So according to your need, you can choose either need to go for GST or need to go for his purification system. This is very simple setup, and also after purification, it cleans the machine cleans itself the purification every column, so you don't have to worry about these things. So the buffer itself will do everything once you set up, and it's ready to use. I'm going to show you some of the examples using the BioRad. This is the demonstration things coming from the BioRad. So these are this initial screen when you're going to set up the samples. It will ask you how you're going to purify the protein, which tag you're going to use. This is here you're going to use the native IMAC, that is his, and then IMAC plus desalting, and then denaturing things. You have the GST option, GST desalting. You have 1 ml one sample, you have two samples, and you have cartridge for 1 ml and for 5 ml. So normally we 1 ml cartridge chromatography is enough for our to get the protein in milligram amounts. Normally we use the 1 ml sample. So just select whatever the uh, experiment purpose for you, and then you go to the next one. Here at the buffer selection, when you go to the different step, the machine will ask you what kind of buffer you need and how much buffer you need. Here you show the different buffer from B1 to B8, and number of sample. How many sample you're going to use is in one sample or two samples. If you have one sample, you just select one and If you have two, just select two. And if you use one sample, there is some illusion, different fraction tubes, like 1A, 1B, 1C, D. 1A is the flow through, B is the wash 1, C wash 2, and then D is the your eluted samples. So if you don't use another one, just leave this thing, machine will, don't say anything, just leave this thing blank. And you have the waste collection here, and you need to make sure that on the other side you have enough water to get the buffer in a required dilution. So next, the machine will ask you to feed the column, chromatographic column, and the desalting cartridge. So this is the column place, and this is the place for desalting things. And there are different steps, but if you have used this column before, the machine will ask you, have you used this machine, or is this a new one? Chromatograph column is a new one. If it's a new one, you have to do some additional steps like showing is here. And then if you used it before, just omit this one and it will go direct you to the next steps. And this is just when you start the machine, it will show you the run is in process and how much time, how much is the progress. It has showing here is the 3% progress. What is the current state in C2? It means sample loading, time remaining you have, expected time of sample to elute, and finally the you will see the elution and then amount of protein in the eluted samples. It will show you. This is just the illusion showing you different one. Just this is the different flow through, and this is your affinity peak, and this is after desalting one. And this section showing the UV, and this is the run volume and time, and this is UV to your concentration of protein in the samples. So this, I'm going to show you different proteins that I have used to purify using this machine. Number one is the West Nile virus, glycoprotein GE domain 3, and then another one is armadillo CD protein. So I'm going to show you at first the West Nile GE protein. This is the sequence of the West Nile GE protein. We have, this is the green one is the West Nile GE domain 3, and then this one is the CD40, Econ CD40 ligand. So we join this together and then make one gene and clone into expression vector, purify it, and then use for immunization. So we have the two different sites here. One is the ECO and then ZO1 linkers. This is the vector that I used to express and purify the protein, and West Nile GE and Econ CD40, and we use this multiple cloning sites. It has GST and his tag at the both end. The 5 prime, this is his, and this is the GST tag. So we can use this protein to purify with the GST purification system as well as with his purification system. So now the steps in cloning of this West Nile GE into this vector. Since we have two different genes, we have to combine it together. So we do it. West Nile GE is in one vector, and then CD40 is another vector. So we use gene-specific primer to purify these two gene, and then we combine, mix an allele, and then do another PCR, and then clone into this vector. So once we clone it, we need to see the expression of this protein in E. coli. Showing here, I'm showing the two different E. coli strain just to see their expression level and their solubility. So this, using the BL21 Fly S, I use two clone, and this is uninduced cell lysate. We don't see any kind of protein expression. And in the In the induced one, this is the clear lysate and this is the pellet. It means the protein going to the insoluble form is forming the inclusion body. And the another clone is also the same. It is also the same. This is going in the inclusion body. Using BL21 star D3, there is another strain of E. coli. Also, the protein was expressed in not in the uninduced one, but it expressed in the pellet. It means that the protein is insoluble and forming the inclusion body. So, and this is just induced at 37 degrees centigrade and induction with 1 millimolar IPTG for 3 hours, and we have enough protein in just after 3 hours. So since this is an insoluble form, so I have to treat in different way just to solubilize the protein, get its soluble form, and then purify it. This is the Profinia chromatogram showing the purification steps of this Western GE after getting this protein in the soluble form. As you can see here, this is the flow through wash, and then I have a nice peak here. This is my protein, eluted protein, and this is a good amount of protein. And here this is, you see the time from the beginning to the end, approximately 1.5 hours, and I have the concentration of protein here around 0.1. It means I have milligram amounts of protein here for here. So next I wanted to see the protein purity in gel. So
how did you solubilize it?
I use some buffer and some solubilizing agent like Tritonex, combination of Tritonex, and then I have also other things, Sarcosil.
Is solubilization an issue?
Yeah, solubilization issue. If you don't use the combination of things, it's never going. You're not going to get the protein in soluble form. You will have the protein, even if it is a small protein, most of the protein goes in insoluble form. So you have to treat it in different way, in different buffer, maybe temperature, just play with different things. And finally, some of the system works for me and I got it in the soluble form. Now here I am showing here, this is the uninduced, this is purified protein and doing a commercial blue stain after SD space. This is the uninduced protein and this is my clear lysate and this is my pellet. Before I show you, most of my protein goes into the pellet, not into the clear lysate. This time maybe I got about 50-60% in the clear lysate. I have still my protein in the insoluble form, but I think this is enough to get some protein purified for immunization purpose or for my purpose of my experiment. So here during my wash step, I lose some protein, the flow through and wash, but in the end I have my eluted protein is a nice amount, and I got probably milligrams of protein at the end. I did in BSA concentration and by gel, and then we use this one to immunize the rabbit to get the antibody against this Western GE. And here right side I am showing the Western blot of the purified protein using a GST antibody. Since it has both GST and his, so I use very small amount of protein, the antigen for Western blotting, and then we have the expected size of the protein in here is about 60 or 65 kV. This is 50 and this is 75. So I have the right expected size of my protein. So this one I used for immunization of rabbit. So next I'm going to show you the expression and purification of the Armadillo CD8. This time I use a different vector. It has PET32A from Novogen, and it has different features than the one I used previously. This is the multiple cloning sites of this vector. It has Tricks tag, as Dr. Kousoulas mentioned in his talk previously, using large tag, it is 105 amino acid, and then it has also his tag at the 5' end, his tag at the 3' end. The other important features of this vector is it has a two cleavage site. One is thrombin, and another is enterokinase. You can use either of these or this cleavage site to cleave your protein and get the purified protein only. If you don't need the tag, or for other experimental purpose, you may need a tag. So this is the expression of Amarilla CD8 into PET32. I also used PGS21 to show the comparison. It has both GST and his, it has Trix and his. This is larger protein because of GST is bigger than Trix, and this is Trix and his, and this is relatively smaller than GST. So I have both of the protein expressed. Again, this is not we can see this is one in, this is the induced one, uninduced. So this is again induced and uninduced. So I have the protein, again this is in the insoluble form, so I have to get it in the soluble form and purify it, and this is the purity of the protein before we do immunization. So this is the different step. Again, this is pellet, this is not the protein, this is my protein here, the bottom one, and I have most of the protein in my clear lysate, and flow through wash I have a minimum amount of protein, but in elution I have good amount of protein to immunize the rabbits. So we use this one to immunize the rabbits, and this is the chromatogram again showing the purification steps in flow through wash. And for the IMAX system, that is his, it has two wash. I mean the IB, IC, and I have my eluted protein here. So this protein we use to immunize the rabbit. So after two immunization, we got the sera from the rabbits as well as the pre-immune sera too. And I have the here also West sera. And we use this sera to purify the sera using a spin column. As you can see here, if the unpurified sera, I just use 2 or 3 microliter. So how big? This is a lot of junk here, but if you purify it with elution 1 and elution 2, we have nice 2-band heavy chain and light chain. This is for premium. And for Amarilla CD8, we have 2 rabbit A and rabbit B, and you see this. I have 2 elution E1, E2, E1, E2, and this is nice band. And for West Nile GE, also the same thing, elution 1 and elution 2. And this is just after purification. We just run a couple of microliters to SDA space and stain with CVV and just seeing the band here. This is from a spin column. So we have a lot of antibodies there, but still maybe not be 100% pure. But we can see at least better than unpurified one. And we have clear heavy chain and light chain band. And this is the one Western blot using the one of the sera from Armadillo CD8 to see the specificity of the sera we got from the CD8 rabbit, immunized CD8 rabbit. Here, if you see this is CD8, and then as a control we use the West Nile GE. This sera is not reacting with the West Nile GE, but is reacting with CD8. At this point, we don't know is it specific for CD8 or we have the tag. Maybe we need to do some next couple of experiment in order to confirm that one. But at least we have the specific band with the antigen from the sera from CD8 immunized rabbit, but not from the West Nile one. So the conclusion here, the Profinia protein purification using this system is fast and very convenient. This system has an informative touch screen display showing each step of the purification. And purification is simple, automated and efficient, and it is very quick, easy setup for purification. Once you have the protein is in the soluble form, then you can play with it. Otherwise, it is difficult to use also and play also with the protein. Thank you very much.